Hello, everyone. We are very excited to be part of the first annual CDRC meeting and hope that you find our session on sarcoma drug repurposing to be a very informative session. My name is Leslie Doris, and I am a pediatric oncologist. I am the acting team leader for the sarcoma team, and I also am the sarcoma scientific liaison. My colleague, Sonia Singh, is also a pediatric oncologist, and she is a reviewer on the rare tumor team, and we will be the moderators for this session. So we are very thankful and excited to have a great panelists and speakers that will be joining us for this. These folks are experts in sarcoma treatment and drug development, patient advocacy, especially in sarcoma, drug repurposing, and developing and running registries for rare tumors. I certainly hope that you all will take the time to listen to each one of these speakers' presentations before we have the discussion session. Some of the big discussion topics that we're going to be including in this is on the use of repurposed drugs in sarcoma, the patient perspective on drug repurposing in sarcoma, which is very important. We're going to learn about registries for rare tumors, and then we are going to end with a discussion on the use of propranolol in patients with angiosarcoma. Given the wide diversity of the audience, I really wanted to start with just a very high-level overview of sarcoma to start with. And then over the next several slides, I wanted to highlight some of the important points about sarcoma that really tend to illustrate why the tumor type was chosen as a pilot for the Rare Oncology CDRC program for drug repurposing. So sarcomas, which maybe some of you have not even heard of, this is a group of very, very rare group of tumors. They are also extremely complex group with unique histologies and molecular alterations that across all the different subtypes. These tumors are tumors that tend to arise from mesenchymal cells, and what this means is that they can mature into striated skeletal and smooth muscle, adipose and fibrous tissue, bone and cartilage, as well as many other different tumor types. So imagine this tumor type can really occur anywhere. There are estimated to be just over 13,000 new cases diagnosed in the United States in 2021. That is a very small percentage and accounts for only 0.7% of all new cancer diagnoses in the United States. Just to put that in context for people who aren't familiar with the incidence of some of the more common types of tumor, breast cancer and lung cancer have over 250,000 diagnoses made per year annually. A unique feature about sarcoma is that within this small total number of patients with sarcoma, there are also over 100 different histologic subtypes, which, as you might imagine, can make drug development and evaluation quite challenging. For patients that have sarcoma, the 5-year survival from the time of diagnosis for all patients is about 65%. And that number includes not only patients with localized disease, but patients that have advanced and metastatic disease. And as you might imagine, this number, the survival number drops quite significantly when we're looking at patients that have metastatic disease, which drops to approximately 16% for survival at 5 years. As you can see from this table of approved drugs, there really have not been a lot of drugs approved since 1974 for sarcomas, and even fewer drugs that have been approved for certain subtypes of sarcoma. Doxorubicin, which most of us are probably familiar with, was the first drug approved. And this drug approved was for an all-comer population, which means any type of soft tissue sarcoma. And this was approved in 1974. The next approval did not occur for almost 35 years. This drug was pazopanib. And this again was approved for the treatment of any type of soft tissue sarcoma, including any of the different subtypes. They had only had to receive just one prior line of therapy. So as you can imagine, with only 13,000 cases of sarcoma diagnosed annually, you can really begin to understand why earlier trials enrolled patients with any subtype into their trials, because it would be nearly impossible to enroll some of these smaller subsets as a single trial. However, the big concern that we notice when you're enrolling patients with any type of soft tissue sarcoma is that it's difficult to, how is it that you assess if one specific subtype or subtypes are actually accounting for all the anti-tumor activity that you see in the results of a trial. And really, for those extremely rare subtypes that may have enrolled, 1, 2, 3, or even, 5 patients, the data that we obtain from those numbers of patients would certainly not allow us to make specific conclusions about the response to that therapy. So then we really started to see a shift in study designs for sarcoma. Investigators and pharmaceutical companies began to evaluate specific types of sarcoma in their trials, so not including all subtypes as had been historically done. This evolution in trial design led to the approval of 2 of the more common subtypes of sarcoma, liposarcoma and leiosarcoma, and giant cell tumor of the bone. Now we have drug approvals for some more of the rarer subtypes, which include tensonovial giant cell tumor, epithelioid sarcoma, and GIST, just to name a few. So thankfully for our patients, science has come a very long way since the approval of the first few drugs for sarcoma, with an increase in not only the number of studies that are being performed in sarcoma, but data that we have gained from small retrospective studies or case series and natural history studies. We have discovered that not all subtypes of sarcoma respond to standard treatment regimens the same. Some types respond very well to radiation therapy, while others do not. Other types of sarcomas are extremely sensitive to chemotherapy, and then others are not. But that's not really one of the most important discoveries we've made. The most important thing that we have learned in the last several years is the discovery in sarcoma and the advances in molecular and genetic profiling in these tumor types. With this, we now have an improved arsenal of data at our disposal for molecular and genetic alterations and natural history data that is constantly growing and evolving. This information is really critical to help design trials that could have a significantly better chance of demonstrating clinically meaningful outcomes for our patients, especially those that have been most often forgotten, that have the rarest of the rare subtypes. This paradigm of one size fits all no longer works when we are evaluating drugs in sarcoma. Having just given you an overview of sarcoma and talked about the handful of drugs that we have seen get approved in the past 50 years, you really can begin to understand the challenges that come with sarcoma drug development and the need to start to think outside the box on how we can facilitate other ways to evaluate drugs that may be viable options for our patients with sarcoma, especially those rare subtypes that have even less than 100 cases diagnosed annually. How can we even enroll sufficient number of patients in trials to make adequate risk-benefit decisions when we're talking about subtypes that have 30, 50, 100 cases diagnosed a year? With such rare diseases, where do physicians go to look for treatment options for some of these subtypes? Looking probably and asking colleagues, and these colleagues may or may not have treated patients, and perhaps if they treated them, it could be just one or two patients. You can look in the literature hoping to find a case report or two on the treatment that is used. So it to me is obvious that clinical trials are still going to be the cornerstone of drug evaluation. But what adjunct to this standard path would be feasible, productive, and provide clinically meaningful data to patients with these unmet medical needs? Are there other ways to overcome these challenges for rare diseases that often lack natural history data, have a significant number of distinct subtypes, which are often histologically unique with different molecular and genetic alterations? And what incentive is there to study a drug for such rare diseases? Is this a role that could be fulfilled by the use of real-world data and evidence? We think there is an answer, and that is looking at drug repurposing as yet another means or another tool to be able to identify potential drugs for our patients. Drug repurposing is a potential way we can evaluate drugs, oncologic or non-oncologic drugs, that may actually be beneficial to our sarcoma patients. With the many challenges in drug development and limited treatment options in sarcoma, sarcoma really seemed to be an ideal candidate with which to pilot the goals and objectives of the CDRC program for rare oncologic diseases. Sarcomas, clearly, as I've stated a number of times, are rare tumors, and they have a high unmet medical need. There is little to no drug development in many of the rarer subtypes. However, and thankfully, we are starting to see an increase in that. We have the opportunity with our collaboration with the CDRC to potentially overcome some of these hurdles. The sarcoma community of physicians and advocacy groups is small but strong and very dedicated. With collaborative efforts, access to natural history studies and registries, and using real-world data, the sarcoma community, and hopefully more rare tumor types, will have another instrument with which to provide a way to get potentially active drugs to our patients. The initiative started by the CDRC is a critical component of bringing drugs to our patients, and we can't thank them enough for their efforts. Our next series of talks will go into more detail on these topics. I would really like to thank Sonia Singh, my colleague in this, my co-moderator as well, and all of the speakers in the panelists, Denise Franke, Dr. Schultz, Tapp, and Wilkie, and Dr. Panziarka. Most importantly, I would like to thank all of the members of the CDRC who have made this possible and have allowed us to participate in this meeting. Thank you so much.